Good morning, everyone. I'm Aaron Guman. I'm a virtual, obviously, volunteer here from NOFA, Vermont, zooming in from frozen Calais in north central Vermont this morning. Welcome, everyone, to the presentation, Planting Garlic into Standing Cover Crops, a UMass research update. This morning, we have Alexa Smychkovich, PhD candidate at the Stockbridge School of Agriculture at UMass Amherst. I'm personally excited to hear the updates. I work with farmers on climate resilience building on their operations. And so this is, cover cropping and reduced tillage or eliminating tillage is a really exciting topic for me as well. And I'm going to do a quick couple intro slides here. So here we are kicking off the NOFA Mass Winter Conference. Cooperative Foodways Building Our Future Together. We just wanted to do a quick land acknowledgement that the land this conference is being presented on is inhabited by the Nipmuc people prior to European colonization. And I'll drop a link in the chat here for anyone interested in looking for a little bit more information on that. Also, we are highlighting racial equity in the food system. There's a bunch of different resources here. And one place that you can participate is a session this afternoon for anyone there in person from 1 to 2 p.m. There's a White Ally Caucus in the Sheehan Hall dining room and a BIPOC caucus in the Sheehan Hall multipurpose room. There's plenty of staff around to gain more information on that. We wanted to thank just some of our partner level sponsors here who have put together support for the course. And also remind everyone of the auction that is happening there in person and can also be found on the app. And a reminder to fill out your surveys after the course. All right, so that's the intro, and I'm going to turn it over to you, Alexa, to tell us all about your research.
Thank you. All right. Thanks for that introduction, and thanks to everybody for coming. Figure out this. Let's start. Okay. And I think I did this. Okay. So my name is Alexa Smychkovich. I'm a PhD candidate at the Stockbridge School of Agriculture at UMass Amherst. Today, I'm just going to give a research update about one of the projects that my lab is working on. The project is about planting garlic into standing cover crops. This is a project we've been working on since 2020. It's funded by the Massachusetts Department of Agricultural Resources, or MDAR. So just a little bit of background about my lab. This is the Hashemi lab, the lab that I work in. That in the middle is Dr. Smychkovich, my advisor. He's also the team leader for the crop, dairy, livestock, and equine extension team at UMass. And that's all of us at our research station in South Deerfield, Massachusetts. So we have a big research farm that we do a lot of research projects on. There's also a UMass student-run farm that works in those fields, and the other extension teams run other projects as well. So the research goals of our team primarily are to enhance sustainability of farming by integrating cover crops into diversified cropping systems to enhance nutrient recovery and recycling, weed suppression, and improve general soil health. So we work in a variety of different systems in order to do this. In particular, our research interests include integrating cover crops into vegetable production, grazing, and feed systems. So we've worked on integrating fava bean into kohlrabi production. We work on saffron production and upgrading cover crops into saffron. I don't personally work on that, but other members of my lab group do. We also work on dual purpose cover crops. So those are cover crops that are grown not just as cover crops, but for some other purpose as well. For example, as a forage, or could be also used as a cash crop. For instance, fava bean can be used as cash cover crop. We've also done work with forage cover crops, or moving cover crops to use as forage, forage. Introduction of cover crops to the Northeast, so trying cover crops that maybe are used other places, but not super popular here, to see if they're feasible to use as cover crops here. Living mulch, tillage reduction, and for soil health and natural fertility, finding ways to improve soil health and fertility through the use of cover crops. So just as a review, cover crops are crops that are planted when soil would otherwise be bare, and they're planted primarily for their ecosystem or their ecological benefits, rather than for their economic harvest. So not for their short-term economic benefits, but rather for long-term ecological benefits. Those can include, alleviating compaction, adding soil organic matter to the soil, reducing erosion, both through the roots and through the residues of annual cover crops, adding nitrogen to the soil, things like that. So things that, oh, sorry, go ahead.
Sorry, Alex, we are not seeing the slides online.
Oh, you're not seeing the slides, okay.
Yeah, is there?
Oh, you know what? I know why, because I didn't do screen share. Do I have to do that?
Yeah, I think you could do screen share. There may be a way to move the camera around, or you could send me the slides and I could share them simultaneously from my computer.
So I don't think I can do screen share because, can you enable participant screen sharing?
Definitely.
That might be easier than, I can send them to you too. The only thing is I have some figures that I think you need to access another doc. I don't know. They might not, not work on your computer unless I send you a bunch of stuff.
Sure, screen share should be enabled.
Okay.
There we go. Perfect.
Is that okay? Is that working?
Yep.
Okay. All right. So
thank you. Just
for the people on Zoom, there's a nice picture of my lab. And this is the slide that we were on. So some things to consider when integrating cover crops into production systems, and these are things that we consider when we're looking at systems that we might want to try to integrate cover crops into. One, are anticipated benefits. So that's, again, important for cover crop species selection. Is your goal to increase nitrogen? Is it to alleviate compaction? Is it to keep the soil protected from erosion during a period of time where you might have rainfall? Is it to capture nutrients in the winter so that they don't, for instance, so that nitrate doesn't leach over the winter and it can be recycled in fall and spring? Field history has this, what is the history of the field in terms of cover crop use or tillage? Again, production cycle and timing kind of goes along with the benefits. This is really important for when you're trying to think about the benefits you want from cover crops, especially if you're looking for nutrient benefits, because synchrony is so important. Just because a cover crop is in your field and it's decomposing and it's releasing some nutrients doesn't necessarily mean that your next crop is going to be taking them up. And I think that can be particularly tricky. So really trying to figure out the timing of when is your crop going to be growing, when is it going to be taking up nitrogen, and when are your cover crops going to be decomposing? And furthermore, what's going to happen to the nitrogen once the cover crops decompose? Is it going to be immediately available to your next crop, or is there going to be some sort of lag where it's immobilized for some period? Termination. So are you going to be able to get into the field to terminate cover crops if they're perennial? Or is it something, are you looking to not have to mechanically terminate your cover crops? In which case, when are you planting them? How are they going to be terminated? Are they going to be winter filled? And residue decomposition and nutrient release, which kind of goes back to production cycle and timing. How is the decomposition of your residues going to impact your production system? Which brings me to the project that I'm going to be talking about today, which is using cover crops to enhance soil health during garlic production in Massachusetts. So this is a project I think I mentioned before. We started in 2020 with funds from the Massachusetts Department of Agriculture. And since then, we've also received an additional grant from Northeast to do some additional data analysis. So in the Northeast, a majority of vegetable producers grow garlic for fresh market. And so since most of you grow garlic, I think you're familiar with the growing season. It's a relatively long growing season. So garlic is planted in October and then it's harvested in July. So that's 9 months. It's a heavy feeder, it requires a lot of nitrogen inputs. And I think the most important thing for our lab, which is why we decided to choose this system as a system to integrate cover crops into, is that it's a missed opportunity for cover cropping. Because many people choose to plant cover crops in the late summer or early fall and let those cover crops, either overwinter or be winter killed, but that provides a necessary ecological benefit in the Northeast is cover cropping in the fall. And if you have garlic in the field, you're missing an opportunity to do that. And as I mentioned before, it's a heavy feeder, so it's taking up a lot of nutrients during that time. And basically we're thinking, is there a way that we can build soil health and increase resilience without taking land out of production in this system? So I don't know if I can, Thank you. I'm like a total Zoom novice. Okay, so some of the drawbacks of the current production models. So passive mulch, so either straw mulch or plastic mulch, cannot feed soil microbes or enhance soil organic matter or recycle nutrients. So one of the benefits of cover crops, especially when planted in the fall or late summer, is that they take up any residual nutrients that are left over in your field and they basically assimilate them into their biomass. And then if they are killed or terminated the next spring, then as they decompose, those are re-released back into your field. So if you have residual nitrogen in your field, it's actually being recycled. It's not being lost to the environment. If you have passive mulch, if you're using passive mulch for your garlic, you're kind of missing the opportunity to take advantage of those benefits. Another big thing is that you're not feeding soil microbes actively when you're using passive mulch. So about 30% of plant photosynthase go into the soil to feed soil microbes. And so that's a very large amount of photosynthase. And so those microbes rely on carbon from plant root exudates as a primary food source. So if you can have as many plant roots in the ground as you can until the frost, then you're actually enhancing your soil microbiome for a long period of time. If you just have passive mulch and just maybe garlic roots are beginning to develop, but they're not very big at that time, then again, you're not providing as much of a food source for your soil microbes. And again, garlic is a heavy nitrogen feeder, so it requires significant fertilizer inputs in the spring. So one thing we were curious about is if we plant garlic into standing cover crop residues, can those residues, decomposing residues in the spring, actually recycle some of the nutrients back to garlic and reduce our fertilizer inputs? And so the system we proposed is to incorporate annual cover crops in our arbuscular mycorrhizal fungi into the garlic production system. I'm not going to talk about the arbuscular mycorrhizal fungi today, but if you're interested in that, you can get in touch with me and I can share my results with you. It was just too much to include into one presentation. So we propose that we plant annual cover crops in the late summer, early fall, and then we plant the garlic into the standing cover crops and inoculate with arbuscular mycorrhizal fungi from the planting. We wanted to choose a commercially available arbuscular mycorrhizal fungi to basically see if it was feasible or worth it to spend the money on those commercially available products. And so we chose one that we could just purchase at the store or order online. And okay, so some of the benefits we anticipated to see was that we would increase soil health and natural soil fertility. Oh, go ahead.
In the previous slide, you said inoculate with something.
Oh, arbuscular mycorrhizal fungi. Oh, so okay. Arbuscular mycorrhizal fungi is a type. Oh, you know what it is?
Maybe I haven't seen the
Oh, AMF. Oh, sorry. I should have defined it. I should have written out arbuscular mycorrhizal fungi. But yeah, the AMF is just
checking.
Yeah. Yeah, you're thinking the same thing. And we chose just like a 4-species blend. Again, I'm not going to really talk about those results today, but just it was part of the project. I wanted to mention it in case anyone does want to talk about it later. Okay, so some of the benefits that we anticipated to see was that we would increase soil health and natural soil fertility with the system, minimize soil erosion, again, not only from the residues in the fall, but also from the living roots in the fall, because we have living roots in the soil holding the soil together for a longer period of time. Feed soil microbes in the fall through the root exudates, and in the spring through the decomposing residues. So as soon as the microbes are active, they already have a food source in the decomposing residues. And of course, living plants draw atmospheric carbon to the soil, so we can sequester carbon and use the cover crops to recycle nutrients to growing garlic, as I mentioned before. So this is kind of the planting pattern. You have the little green things there. Imagine those are cover crops, and we're planting. They're standing, they're living, they're going to die in the winter, and we're planting the garlic in October into the standing residues. So here's just a brief timeline.
So there's mustards.
This is actually a cover crop mix of pea, oat, forage radish, and I think there might be something else in there. This is there is definitely radish in there. Oat, I think it might just be oat, peas, and forage radish. This was a commercially available mix that was donated to us. And so this is just a timeline of what the project looked like. So in September, or you'll see in the second year, we planted it in late August. We planted our cover crops, let them grow. In October, we actually hand planted the garlic into the cover crops. In November and December, the cover crops are starting to winter kill. By March, the residues are decomposing. April through June, the garlic is growing through the residues. And in July, we're harvesting the garlic and we're separating it for analysis. Sorry.
In the October project, have you left? Have you left row space where you're going to put the garlic?
Yeah, so we planted the cover crops in rows, and then we basically used, like, actually in here you can see we planted the cover crops in rows, and then we planted the garlic right in between the rows. So the spacing between the cover crops is 7.5 inches. So the garlic too is about 7.5 inches apart.
So you're not planting directly in the cover, you're planting in the rows between the cover?
Yeah, yeah, exactly. So we're planting into rows in between the cover. The cover crops are planted in rows that are 7.5 inches apart. And so then you have this pattern where you have, after you plant the garlic, you have cover crop, garlic row, cover crop, garlic row. But the cover crops are actively growing, and the garlic is getting established and starting to produce fruit, but it's not going to sprout until the following spring. So we're hoping that we're going to minimize competition, but you'll see when we get the results if that actually happened or not.
So do you drill the clothes in, or do you have a furrow that you're putting them in?
So we've tried both. So we used we drilled them in with like a no-till dibble first with a dibbler, and then we also tried doing it with a furrow, just like made a furrow with a hoe and put them in, and they both worked okay. So that's how we did it, just for the.
And what about weeds?
I'll get to that.
Hey, Alex. Alex?
Yeah.
We had a request, if you could just go back to the slide and just run through the timeline real
More
quick.
slowly and more loudly. Got it. Okay, I can be louder.
Thank you very much. It's also for the folks online, it's loudest when you're by the laptop here.
Okay, all right. I'll try to stand still and I'll try to talk loud.
Thank you.
All right, so timeline. All right, so September, we planted the cover crops. 1 year we planted early August, and I'll talk about that a little bit later too. And so from September to about November, December, the cover crops are actively growing. And so you'll see in the second two photos, they've put on some biomass. And so you have this like large amount of cover crop in the field. And then in October, about mid-October, we went through and between the rows of cover crops, we planted the garlic. And so we used a no-till dibbler for some of we tried to do it with a no-till dibbler. We tried to make furrows with a hoe. As you'll see later, we actually also used a mechanical no-till transplanter, and all three of those worked okay. And then by November, December, you'll see that the field is starting to winter kill. The cover crops are dead, and they're going to start decomposing once the weather warms up. So in March, you can see these are already residues. And so those are the residues that are going to decompose come April when the garlic sprouts. And then from April to July, the garlic is growing. And then in July, we're harvesting the garlic. And then what you see in that very last photo. So again, this is a research plot. So you see they're all labeled by plot number there. And then in the last photo, you see We actually, after harvesting the garlic and taking yield data, we also separated it into roots, bulbs, and aerial parts. So we could do different analysis on all of the different parts of the garlic to see where is the garlic storing nitrogen, what is the nutrient content of the bulbs, and things like that. So we actually had to separate all of the garlic into various parts to do that. So that's what you see in that very last part. And then, of course, in the winter, we do all that lab work that's not on this timeline. So I hope, let me just open the chat. Oh, sorry, I don't know where that is. But hopefully that was.
Did you use different varieties or is that?
This is all one, that's a good question.
German, right?
It's German extra hardy. And the reason we use one variety is because we want to keep it consistent. Yeah, we didn't want any variation between the plots. Okay. All right, so these are the cover crops that we chose and some of the reasons that we chose them. We have 4 cover crop treatments, and we compared this with a no cover crop treatment as a control, which we mulched with straw. So all of these cover crops have different carbon to nitrogen ratios. I'll talk a little bit more about that later. So the 3 cover crops that we planted in monoculture are field pea. Field pea is a legume, so it fixes atmospheric nitrogen, which is why we chose it. We wanted to include a legume crop. It's also relatively cold hardy, so we knew it could probably produce a lot of biomass before it was winter killed. It's a high biomass crop and it has a low carbon to nitrogen ratio. So again, I'll talk about that more in a bit, but that's indicative that it'll decompose relatively quickly. Oat is fast growing. It's good for suppressing weeds. It has a lot of biomass. It has a pretty robust root structure. And also it has allelopathic effects that may inhibit weed growth. It's good at recycling nutrients to successor crops. It's good at controlling erosion, again, because of that root system and that high biomass that has a higher lignin content, and it has a high carbon to nitrogen ratio. And then the last cover crop that we chose was forage radish. That's because it's a biodrilling cover crop, so it can alleviate compaction. Again, it recycles nutrients and it goes pretty far down into the soil profile, so it can recycle nutrients from further deep down in the soil. It's quick to establish, it adds significant soil organic matter, and it has a low carbon to nitrogen ratio. And then our last treatment, a cover crop treatment, was a mixture of oat, forage radish, and field pea. This mixture combines, so cover crop mixtures combine the benefits of individual cover crops. They increase biodiversity, and also they create this intermediate or balanced carbon to nitrogen ratio. So if you have low carbon to nitrogen ratio crops and high carbon to nitrogen ratio crops, it kind of creates this more balanced and perhaps a more balanced decomposition scenario. So as I mentioned, we planted by hand, and also we tried to use the furrows. We also wanted to try it on a larger scale. So we used a no-till transplanter in a field that was cover crop just to see if it would work. And this is actually a video, so I'll show you what that looks like.
That's October or so?
This is October, yeah. So it's the same, this is the same time. And we just did this to see if we could scale this operation up. This wasn't really part of the research.
It's able to do 3 or 4.
It does 2 rows at a time, yeah. But it goes much faster, as you can imagine. And it was with this amount of residue and for this purpose, it worked really well. So. Mm-hmm. Oh, okay. So data collection. So what did we actually look at? In addition to decomposition, which I'll talk extensively about, we looked at soil health and nutrient data. So we looked at microbial respiration in the soil while the cover crops are decomposing. We looked at soil food web dynamics. I'm actually still doing the data analysis on that, so I won't be able to provide you with results on that today. Active carbon, that's a new thing that we're going to look at. We just got a grant from Northeast SARE this past year. So in the next few months, we'll be running active carbon on the soil samples during decomposition. So for those of you who aren't familiar, active carbon is a soil health indicator, and it can monitor short-term changes in soil. And it's the portion of soil carbon that is can be readily used by microbes as a food source. So that's, again, we're gonna have results on that in the next few months. looked at soil nitrate, and then after the 2 years, we did an earthworm count and looked at compaction, just to see if there were changes after 2 years of running the system and those parameters. For the garlic, we looked at yield. We looked at the nutrient density in the bulbs to see if the quality of garlic changed. We looked at the growth curve, and that was important because we wanted to see how the garlic was growing, what the curve looked like in the spring, and to compare it to the decomposition of the cover crops to see if there was synchrony between nutrient uptake and garlic growth. Chlorophyll content, which is used kind of as a proxy to see if the garlic, while it's growing, has enough nitrogen. Fertilizer recovery, nitrogen use efficiency. I've mentioned those before. Again, we're actually still doing those in the lab, so I'm not going to present on those today. So now I'm getting to the results. So the first thing I wanted to talk about was the cover crop biomass yield. So we planted the cover crops 2 years in a row, and how much biomass did we get from each cover crop? How much nitrogen was in that biomass? And the first thing I want you to pay attention to is that we planted the cover crops 1 week apart on both years. We planted a week earlier the second year than in the first year. And if you look at this is dry biomass pounds per acre, but if you look at the biomass differences between 2020 and 2021, and again, those translate to nitrogen differences, they're huge. So there's 2 things that you can do to change the biomass of a cover crop. There's 2 things that control that. One is seeding rate, and the other is planting date. And so basically, we use the same seeding rate, but we altered the planting date by just 1 week, and we saw dramatic differences. I think that's important, especially in the fall, because as temperatures start to decrease, the longer you wait to plant cover crops, you're going to see yield penalties. And of course, through the yield penalties, you will also see nitrogen penalties. Yeah, do you have a question?
How do you factor in and what will see significant seemingly warmer term that we're experiencing?
Oh, like you mean as temperatures get warmer, how can? Well, I think what that does mean is I think you can maybe, and I hesitate to say this because there's been research that shows, I think the cutoff date is September 15th. You should plant your cover crops by September 15th. I think we are probably going to see that you can plant cover crops later as temperatures get warmer, and also as frosts are delayed. You know, where if we see colder temperature kind of recede and we're not getting cold snaps as early, then yeah, maybe there's going to be a change in when you can plant your cover crops. But as of now, I think, yeah, still late summer is better than early fall. And if you're planting early fall, try to get them in before September 15th. And so, yeah, so I just wanted to point this out too, not only because I've heard that before, especially being in my lab and talking to my advisor who really drills this home like all the time, that you have to plant your cover crops early, and then even just for me, just for personal experience, seeing this dramatic difference of actually looking at the yield in a week of difference when we planted 1 week apart. And of course, there's also weather differences. These 2 years were different in weather, I'm sure. So I'm sure, you know, this isn't in the same year, so that's something to take into account. But another reason to pay attention to this is when you're thinking about decomposition from both of these years, you're having a lot more biomass decompose in the second year than in the first year. So again, this is just a visual. This is October 2021. You can kind of see where the cover crops are in terms of height and biomass. And then this is October. Oh, this is wrong, but this is October 2020. Sorry, this is October 2021. 2022 was last year, so there was nothing there. So this is, but yeah, here are the 2 differences: year 1, year 2.
This is the earlier planting, right? Year 2.
Yeah, this is the earlier planting that we planted in late August. Okay, so now weed pressure. So one of the things we wanted to look at, and we was part of this grant, is to look at can we eliminate the need for mulch altogether with cover crop residues? And the answer is no. You should not do that unless you want to hand weed, because this is what, there's just no way around it. The weed pressure gets it gets ugly. So this is March 2021. So the first year we hand weeded because we didn't want any yield differences to come from weed pressure. So we diligently hand weeded. It took us many, many hours. And then the second year we actually we mulched with straw in the spring. So this is April. They're starting to get established. And here's June. And so this isn't a research plot. This is a plot that we let go to the weeds to see how bad it would get. So just so that you don't, if you look at yield differences, you're not like, oh, well, that looks terrible, you know? So, okay. So yeah, the answer there is there needs to be some sort of either hand weeding or mulching going on to use the system. The residues are not going to keep the weeds down in any of the cover crop treatments, even oats. Okay, so now moving on to garlic quality.
That's a good question.
Yeah, go ahead.
Would you say the this cover cropping system addresses soil health and nutrition more than weed suppression?
Yeah, I'll talk about all the different things that it addresses. And I think I'll get to this, but I think the bottom line, well, yeah, I'll get to this. I think that really it comes down to ecological benefits and more like long-term visions for your farm than immediate benefits. Yeah. It's not going to be this like magical thing that now you're going to get like, this is going to be the best garlic crop you've ever seen, and you're going to not ever have to mulch again and never grow garlic the way that you were growing it before. I think it's an option, and there's reasons to go with this option or not go with this option.
I just had a quick question.
Oh, sorry. And there's also, I'll take the Zoom question first, and then I'll, sorry, go ahead. I can't see.
Yeah, I was just curious, are these slides going to be available for review later?
So, probably not immediately. The only reason is a lot of this is unpublished data. So I don't think I can share it until I publish it. But after that, they can be available, yes. Which should be hopefully in the next 6 months, if all goes according to plan. But also you can get in touch with me. If you have specific things that you want to review or you want me to send you specific slides, you can just get in touch with me. I'll have my email on the last slide, and feel free to reach out to me with anything that you want to talk about.
Thank you so much.
Okay, so garlic quality. So I think. Oh, sorry, you had your question too. Sorry.
Have you looked into flame weeding?
I haven't. That's a good thing to think about.
You can at some stage flame weed slide, but I don't know whether it.
Yeah.
I think that's something.
Yeah, I'll talk to you.
It's just like flaming weed.
Yeah.
At some stage garlic weed, you can flame weed it. I haven't done it, but I've read about it. I think it's that.
Yeah, I think that's a good point. I think that like if you were serious about adopting the system and figuring out the kinks, like there are a lot of ways that you could make it better and make it work in an operation. Again, like we only had 2 years and so many things to do, so yeah, we couldn't look at everything, but yeah, I think that that's a good point. I mean, it might be something to consider for dealing with weeds, because I think that that's one of the bigger problems with the system is weed control. Okay, so for the garlic quality, we actually looked at the nutrient density in the bulbs. And the way that we did this is we took some samples of the bulbs from every single treatment. We chopped them up, we dried them, we ground them, we turned them into ash, we suspended them in a solution, and then we ran them through this microwave plasma, and that way we could actually see the nutrient content of the bulbs in each of these treatments. And so we wanted to see if the species of cover crops impacted the nutrient density in garlic. We actually looked at more nutrients than this, but these are the only nutrients where we saw significant differences. And so as you can see, there's not huge differences. If there's the same letter on any of the bars that have the same letter, that means there's no statistically significant differences among those treatments. And if the letters are different, then there are. So if you're looking at potassium, the only real big difference that we see is that the mixed cover crop species has the garlic that was planted into the mixed cover crops has accumulated less potassium in the bulbs than for instance, the no cover crop treatment. Yeah.
Remind me, the no cover crop treatment, was that mulch?
It was mulched with straw in the fall. It was mulched in the fall. Mm-hmm. Yeah. So we wanted to keep it as close to garlic production as it is now as possible. Then again, in the phosphorus, the biggest difference is between forage radish and field pea. So we saw reduced phosphorus uptake in the garlic that was planted into forage radish than field pea, and manganese, which is a micronutrient. So you have reduced manganese uptake in the oat plots versus the no cover crop. Everything else, there's no really significant differences between the no cover crop and the other treatments, and there's no significant differences between the oat treatment and, like, for instance, the field pea treatment, but there is between the forage radish and the mixture. And this is, again, just to say, I think the big take home here, at least for me, was that planting cover crops, especially if you're planting cover crops a lot and you're thinking about these systems, they actually do impact the nutrient concentrations of your cash crops, maybe not in big ways or ways that you care about, but if you are thinking more and more about nutrient dynamics on your farm, just know that cover crops take up nutrients differently, and as such, they recycle nutrients differently to successive crops. Again, you might not really care if your garlic has more, less phosphorus in one treatment than the other. It might not matter to you at all, but again, if you're thinking about things like nutrient dynamic, it's just something to consider, that these plants behave differently, and so your system is going to have different inputs depending on how you choose to manage it. Oh, I think it's frozen. Oh, sorry, there's a question.
Yeah, do you know how?
Yeah. No, I'm not sure about the taste in terms of these nutrients. I know that garlic is a heavy phosphorus and potassium feeder, so it needs those nutrients. So I'm guessing that the fact that there's some treatments that it might not be getting as much as other treatments is not a good thing. The only thing that I know significantly impacts the taste of garlic is sulfur, because it's responsible. It's found in the compound allicin, which is what gives, if you all grow garlic, you probably already know it's good. What gives garlic its spice. And so sulfur is an important compound for allicin production. So sulfur, in terms of just taste, is important. And we looked at sulfur and there was no significant differences, which was good. Okay, so now I guess the big thing, garlic yield. And when I saw this, I was discouraged, but then when I thought more about it, I was less discouraged. So hopefully you kind of follow the same emotional journey as I do over the next couple of slides. So we did see a significant yield reduction when you plant garlic into standing cover crops, rather than just planting it into straw mulch and not into standing cover crops.
What are the numbers?
So
on
the y-axis, so this is 8,000, 7,000. These are pounds per acre. So we saw about 6,500 pounds per acre for the cover crops, and around a little bit less than 8,000 pounds per acre for the cover.
And you'll have the garlic numbers.
Sorry, these are the, this might be confusing. This is the garlic numbers. So this is the pounds per acre for garlic in all of these plots. So the no cover crop plot, we got that much garlic, and then these are the other plots.
Last question.
Yeah.
What was the yield of nitrogen plants to get that much garlic per acre?
Oh, this is per acre. So this is adjusted to be per acre. So we planted plots. We planted 60-foot plots, and then we harvested the 60-foot plots, and then extrapolated the data to be per acre.
What was it like?
Yeah. So again, it's not like a good like, we used about 10,000. I think we planted around 10,000 plants total. But again, we planted on like maybe something like a quarter of an acre rather than a full acre, and then we extrapolated the data. Our spacing in row. So it was 7.5 inch rows and then 6-inch spacing in row. Any more questions? Yeah.
So how much you say you did quarter acre plot, you said?
Yeah, about that.
So how much weight did you plant to the quarter acre garlic?
I don't remember, but I have it written down, and I can tell you right after there. That's because
to know, because that's what will tell you what the yield is,
Right. Okay.
If you planted, 4,000 pounds of garlic and you got 8,000 back, that's 2 to 1 yield.
Oh, I see. So you want to see like, what weight did I plant, and then how much they? Okay, yeah. Okay. Yeah, I have the numbers for how much weight we had, so I can. It was, yeah. The spacing was standard, and we used a pretty popular variety, German extra hardy, and so and so then we got the yield from those plots, and yeah. But if you want to talk a bit more specifically about the plots and like how much you know, how much pounds we've planted per plot and all that. I'd be happy to do that after I have it all written down. So, okay. So the possible reasons that we think could be responsible for the reduction in yields are, one, competition between garlic roots and cover crops during establishment in the fall. And so again, this is something that we didn't think would be a big issue because garlic doesn't sprout until the spring. But actually, if you actually pull out a garlic clove in the fall, you'll see it's already developing roots. So it's starting to get established. The one thing to think about there is that you have these cover crops that you're planting late summer, early fall, and they're getting established and they have a lot of biomass by the time they're already established and producing a lot of biomass by the time this garlic is going in. And then this garlic is just trying its best to get established in this like already established field of cover crop. So that's something that there's definitely, if it's producing roots, there's going to be competition. And so there could, that early competition could be resulting in a yield loss, or nitrogen immobilization by the cover crops. I'll talk about that a little bit more in a bit, but yeah, again, sometimes when you have decomposing cover crops, you have to think about the nitrogen dynamics. They're not, the nitrogen that comes out of the cover crops isn't always immediately available to the successor crop. So nitrogen immobilization is common and something to consider too. And so this is just a visual, and then this is just anecdotal. It's like not something that this might not matter that much, but these are the differences between the garlic in the 2 years. And one thing that we really noticed in 2020, or yeah, the first year, 2021 versus 2022, is that the roots in the garlic plants in 2021 were huge. And the second year, when we had actually more cover crop biomass, they were relatively small. Again, I'm not saying that's because there was more competition. It's a thought. It might not be because of that. We also had very different weather patterns. There's a lot of things that go into that. So it was just an observation. Yep.
Was there a significant difference in the size of the pods?
Not in the 2 years. No.
It's beautiful.
Thank you. So one thing I want to talk about, especially when I first saw the numbers, I was like, oh no, I'm getting this huge reduction in yield. And then I remembered about land equivalent ratio. And that I think is something that if you're working in intercropping, it's a really important concept to keep in mind and consider. So the land equivalent ratio is the ratio of the area under sole cropping to the area under intercropping or polyculture needed to give equal amounts of yield at the same management level. So if you increase your land equivalent ratio, you're actually giving yourself a yield advantage. If you decrease it, then you're giving yourself a yield disadvantage. And if it's equal to 1, then you're really not doing yourself, you're not really doing yourself any yield favors by intercropping. And I'll give an example of this in a second. So here's the equation, pretty standard. It's again, the ratio of the intercropped yield to the sole crop. So if you have 2 crops, A and B, then if you have the yield of the intercrop yield of A over the sole crop yield of crop A, added to the intercrop yield of crop B to the sole crop yield of crop, divided by the sole crop yield of B, then you'll get your land equivalent ratio. So if we use the example of intercropping fruit trees with grain, then you have, and say you have your sole yield of grain is 5,000 kilograms per hectare. Your intercropped grain yield of grain is intercropped with the fruit trees is 4,000. So you're getting a decrease seemingly yield of your grain. So that's a 0.8. And then but you're intercropping it with fruit, then you have, say you have a big sole yield of fruit that's 15,000 kilograms per hectare. When you're intercropping, say your yield is decreasing, again, because of this competition for resources, but combine them and you have a land equivalent ratio of 1.4. And basically what that means is that 1 hectare of your mixed planting produces as much yield as 1.4 hectares yield of monoculture. Because one thing you have to remember is that you're combining you're basically combining multiple acres of cropping into 1 acre of cropping. So as long as your land equivalent ratio is going up, then you're actually getting a yield advantage. If your land equivalent ratio is going down, your yield is suffering so much from both crops that your land equivalent ratio is going down, then you're getting a yield disadvantage. So when we're thinking about intercropping, it's more important to look at land equivalent ratio than it is to look at just yield compared to monoculture, because it's not a monoculture and you're planting way more plants in the area. So that's something that I started thinking about with this garlic system. And so it drew me to some conclusions. One, I just talked about this. land use equivalent ratio is a useful tool to assess yield advantages and disadvantages. Two, another thing is if that garlic yield lag, yields drop seems significant, adjusting the seeding rate of cover crops I don't know because we haven't tried this, but it might be a way to increase your garlic yield, is if you decrease the seeding rate or you plant a little bit later, you get less competition, you might be able to increase your garlic yield. Again, it's a thought, hasn't been tested. And again, I think an important thing is to remember what your goals are. So the goal of this project wasn't to increase the garlic yields and get the most garlic that we possibly could. We actually already know how to do that. There's tons of recommendations on how to do that, which fertilizers to apply at what rate, you know, all of that. And so if our goal is to increase the sustainability and resiliency of this system and try to feed our soil microbes to as long as possible, add soil organic matter, then that changes things. And then that yield drop doesn't necessarily look so bad. But that being said, who am I to say that? I mean, if you have an economic plan and you need this yield of garlic, then it's up to everybody to decide how much of a yield decrease they can take for an environmental benefit, which I think leads into a larger discussion of economic versus environmental benefits, because in this case, you don't have two economic cash crops that you're growing together and you're getting this yield advantage of two economic crops that you can both sell and make more money of on your planting with cover crops. So you're kind of looking at these sort of vague long-term benefits, which I think are really important, but they're not necessarily short-term economic benefits. And so that's something to consider when choosing these types of systems, is how much can you afford to basically sacrifice economically for these, yeah, go ahead.
How do you think about the rotation of like garlic and cover crops, and with your experiments, you do it on the same plot between 2020 and 2021? Did you do it on different pieces of land?
No, we did it on the same plot. And the reason that we did it on the same plot, there's disadvantages and advantages. The reason that we did it on the same plot, which is actually uncommon for research, usually you do it, if not on a different plot of land, you at least randomize the design, so you're not getting the same treatment on the same plot of land twice. But the reason we did it twice in the same plot of land is because the history of this field is that it was pretty heavily tilled and it wasn't organically managed. And we wanted to look at some sort of like transitional effects. So after 2 years of, like, for instance, planting oats twice, do you see some sort of soil health benefits? That had less to do with the garlic as it had to do with the cover crops themselves. So like if we plant oats 2 years in a row, are we seeing some type of benefit rather than planting field peas 2 years in a row? For garlic, I mean, that's actually not good practices. You don't want to plant garlic in the same place twice. And we did actually sting us because we got fireworm pressure on one end of the field a little bit the first year, and then it was a lot by the second year. So yeah, that's in terms of just like this system. Yeah, I would not plant garlic in the same year and the same cover crops, but for the sake of our research and some of the other research questions we were asking, we chose to plant them in the same plots in the same place 2 years in a row. Yeah. Yes. Yeah. No, we basically, yeah, we did. We factored in the part. There was one, we have it set up as replications. There was one replication that was heavily affected, and the plots that had wireworm we took into account, and we either didn't count them or we counted healthy bulbs from those plots. But yes, we did factor it in. So it wasn't like the wireworm, this field was affected with wireworm, and then that impacted the yield in some of the, does that make sense? It doesn't. You look at it.
So you set those had wireworm damage.
We didn't count them in the economic yield. No, because you wouldn't sell those bulbs if they were, yeah, you know, if they were small and they were covered in wireworm. If it wasn't something that you could sell, we wouldn't count it in the yield. Okay, so next I'll talk briefly about cover crop decomposition. This is a big part of the project, and they're not going to show all the cover crop decomposition data. But again, this is just, if you remember, the cover crops were planted in the fall, terminated, winter killed in the winter, and then actually decomposed in the spring. So the way that we were able to track decomposition is right about the time of winter kill. We went out into the field and we harvested a bunch of cover crops. These came from different plots so that they weren't interfering with the experiment. And we harvested the cover crops, tons of them. We washed the roots and we separated them into root and aerial parts. And then we put them into, these are called litter bags. These are literally just made out of window screen or mesh. And so we just made these square envelopes of mesh. And we took a known amount of fresh weight for every single cover crop treatment. Again, we replicated this roots and aerial. So we put the roots into one litter bag, we buried them, and then the other aerial, for the aerial parts, we put them on the soil surface to mimic where those residues were going to be. And we made tons of these bags. So we replicated them, and I think, yeah, we made several hundred each year. And then in November, we put them out into the field. So we actually buried the roots of all the different cover crop treatments in the field. And then we put the aerial parts on the surface again, just to mimic where they would actually be during decomposition. And then in the spring, we harvested these cover crop decomposition bags. So we picked them up from the soil surface and dug them up every 2 weeks from April to July. So every 2 weeks we picked them up. We dried them so we could get a dry biomass. And then we also looked at their nitrogen content, their carbon content, their carbon to nitrogen ratio, and their biomass so we could create a decomposition curve. Simultaneously, we dug up garlic, growing garlic, every 2 weeks at the same time. And we separated it into the aerial parts, the roots, and the bulbs. And we tracked the changes of the growing garlic too to see how the garlic was growing during that time, so we could see if there was synchrony between decomposition and garlic growth. Also at the same time, we took soil samples from all of these plots, and I'll talk about this a little bit later, but from the soil samples, we looked at soil nitrate. We're going to look at active carbon, which is that carbon source, the microbial, the part of soil carbon that is can be used for microbes as a food source. And we're also looking at soil food web analysis in those crops, and we looked at microbial respiration in the soil samples. So just a brief review about carbon to nitrogen ratios. Carbon to nitrogen ratios of crop residues influence the decay of the residues. So I don't know if you've heard about the ideal microbial diet, which is variable, but it's 24:1. So basically, I'll talk about this a little bit more in my next slide, but the microbes ideally would like a carbon to nitrogen ratio of their food source to be 24:1. So if you have crops with high carbon to nitrogen ratio, so something that's generally above 20, those are residues that usually have a lot of lignin content, stems, woody residues. Oats generally have a lot of high carbon to nitrogen ratio. And these high carbon to nitrogen ratio crops can lead to, or inputs, can lead to nitrogen immobilization in soils. Low carbon to nitrogen ratios, so things that are usually less than 20:1, they usually have a high proportion of like leafy greens, so cellulose, hemicellulose, and they're likely able to provide nitrogen to systems. They encourage nitrogen mineralization. Just to go a little bit further in that, this is an example. So for every 20, when we say that the ideal microbial diet is 24:1, what we mean is that microbes, for every 24 units of carbon that they consume, they need 1 unit of nitrogen. And so if you have a low carbon to nitrogen ratio, for example, 10:1, then for every 24 units of carbon that microorganisms consume, they receive 2.4 units of nitrogen. So that's more than they need. So because they only need 1 unit of nitrogen for every 24 units of carbon, they excess 1.4 units of nitrogen are released into the soil in a plant available form. And so then the plants can take it up, and that's how we get mineralization. If you have a high carbon to nitrogen ratio, the opposite happens. So you have for every 24 units of carbon, if we're using the example of 80:1, for every 24 units of carbon that microbes consume, they only receive 0.3 units of nitrogen, which is less than they need. And they have to get that nitrogen somewhere. So they look for it in the soil, and then they take up soil nitrogen, and they're competing with plants for that nitrogen, and they can usually outcompete plants for that nitrogen. They're much more efficient at obtaining soil nitrogen than plants are. So that's what we call nitrogen immobilization. And so when nitrogen is immobilized, it's not gone from the soil, it's just incorporated into microbial biomass. So it will be released later, but it's not going to be immediately available. When those microbes die, then that nitrogen will be released back into the soil. And so it basically, it could be more of like a time lapse thing than just that nitrogen is gone. But if you're actively growing a crop, it's important to consider. So one thing, so I'm just going to drink the water, but here are some of the, oh, go ahead.
Maybe off topic, but on the carbon to nitrogen ratio chart, if you look at, like, for example, the progression of like rye, the carbon ratio will go way up as it matures.
Yeah.
What happens with the excess? Is it just building carbon? So let's say after the rye is already mature, you cut it, becomes straw. The excess nitrogen, is it just going into the atmosphere at that point? Because if you look at like the rye cover crop and pieces of mature stage, which 37.1 versus rye straw, where is the excess nitrogen running to?
So that's a good question. I'm not totally sure I can answer it, but I'll do my best. So again, these are ratios. So again, as the rye matures, it's producing more lignin. When it's young, it has more of like the leafy greens, the leaves, so cellulose.
After it's between those two stages.
Oh, I see. Oh, between the, oh, you mean between the rye cover crop and the rye straw? Yeah, or I think there might be some, yeah, I guess I'm not really sure. I think it might be also, maybe that it's like decomposed quickly. And then when you're basically receiving rye as straw, you're just getting this most of the stalks and the lignin. I don't know. I'm not going to pretend like I know the answer. I'll look into it. That's a good question. But yeah. Oh, no. No, now I'm interested and I'm going to spend a lot of time Googling this after, but, okay. So these are some of the results that I got from, as I showed you before, I looked at the aerial and the root decomposition separately because they have different compositions. There are also some are below ground. They're going to decompose at different rates than the aerial, which is on top of the ground. For some of the results, I'll show you the combined, and then for some of them, I'll show you separate. So these are the carbon to nitrogen ratios as these residues are decomposing. So the first point is in November. That's when I actually put the residues out onto the soil. And then April to June is the following year. That's when I started collecting the residue bags. So for the aerial parts, most of the cover crops had aerial carbon to nitrogen ratios that were in the mineralization category. So I don't know if you can see it's a little bit cut off. But anything below, I'd say 20:1 is considered mineralizable. And so all of the aerial parts are in that mineralizable category throughout their decomposition period. So even as they decompose, those carbon to nitrogen ratios remain mineralizable. The root residues are a bit different. So we see that oats, the oat carbon to nitrogen ratio of oats is always so high that it's always going to encourage immobilization dynamics in the soil versus forage radish, that big fleshy root, it's under 20, very low carbon to nitrogen ratio, very prone to mineralization. So that's just something to keep in mind when you look at like the nitrogen trends of these crops too. Forage radish, low carbon to nitrogen ratio the entire time it's decomposing. And so here's just the biomass trends for 2021 and 2022. You'll notice they're different. So again, that first point is November, and then the second point is April 1st. I think that's the first thing I want to draw your attention to. In 2001, from 2020, fall 2020 to 2021, most of the cover crops, only about 20%, less than 20% of the cover crops had decomposed before April 1st, which is when we actually started tracking decomposition every 2 weeks. In 2022, so from the fall of 2021 to the spring of 2022, you actually saw some big differences, with forage radish being the most dramatic, where you saw more than 60% of the aerial parts of forage radish had decomposed by April 1st. And with the other cover crops too, even the mixture, you saw about less than 50%, but more than 40% of the mixture had decomposed before April 1st. That's before your growing season. So that's just something to keep in mind. And for root decomposition, you also saw some differences, not as dramatic as some of the aerial differences, but again, you have a lot of decomposition happening for some of the crops. before April 1st, but again, not a majority of the decomposition. So perhaps more interesting is to look at the nitrogen release from the decomposing cover crop residues and how that changes with the different cover crops from the fall into the spring. So again, the time points are the same. The first time point is November. This is for 2000 I'm sorry, I messed this up. It says November 2021. That's November 2020. I'm just going to repeat that a couple times in case people miss that, so that's not confusing. It's November 2020 that I put those bags in, and then this is the year 2021 for April to June. And so if you look at the nitrogen release, you have a lot of some nitrogen. I'll show a better chart of this on the next slide, but the boxes that you see here, that's the actively growing time of garlic. So this curve is actually made from our data of the garlic growing throughout the season from April 1st to June 23rd. So most of that active growth that you can see is happening from about April 14th to June 10th. So that's when it's taking up a lot of nitrogen. And so that box is kind of, that's the box that you see on the left for nitrogen release for these cover crops. And so if we look at the actual numbers for the nitrogen contributions from these decomposing cover crops for this year, or for 2021, you see that the total percent of the total nitrogen in these crops released before April 1st varies by cover crop. And this is going to vary by year too. But so for oat, you only have 9% of the total nitrogen in the oat released before April 1st, so before your growing season. It's a pretty small amount, about 1.5 pounds per acre. But if you look at the total amount of nitrogen released during the season, 19% of the residues, it's only about 3 pounds per acre too. And that's not going to offset your nitrogen, that's not going to offset your nitrogen fertilization very much either. So similarly for field pea, you see more of the nitrogen released before April 1st. And so more of the nitrogen lost before April 1st. And about 30% of the total nitrogen is released during your active garlic growing season. Again, so only about 12 pounds per acre. Forage radish, we see in both years, we see the most dramatic or the biggest nitrogen contributions from forage radish, which might have to do with that low carbon to nitrogen ratio. And it's being prone to mineralization. So again, you lose 29% before April 1st, that's about 11 pounds per acre, and about 50% of your forage radish, the nitrogen in your forage radish is released between that time period of April 1st to June 23rd. So that's probably the most, I think, for me, interesting result. And then for mixture, you kind of have this intermediate result as well. And so I'll show you the results for 2022 as well. And one thing that I want to look at, and I'm hoping to do next, is actually see if I can find any correlations between the biomass decomposition of these crops and the nitrogen release. The reason I want to do that is because, as you may have noticed from some of these figures, you might have, for instance, 30% of the biomass has decomposed, but 50% of the nitrogen has exited the crop. So I'm trying to see what those relationships look like, so to make it more of a easy visual tool. So if you could just visually see, well, this is how much of my cover crop is left. If there's, so that there's at least some idea of like how much nitrogen may have or may have not been released. So these are the results for the second year. So they're actually very different. So, oh, go ahead.
So are you adding other fertilizers, or is it just depending on the cover crop?
So good question. For this part where I'm looking at the decomposition, I'm only looking at the cover crop. For the actual research project, I have 4 nitrogen levels. So I have 0 nitrogen. I have 33% of the recommended rate, which is 120 pounds per nitrogen per acre, 66 and 100%. And so I'm looking at the yield differences between all of these in the cover crop plots. So I'm trying to see if there's any scenario where we can reduce nitrogen inputs and get similar yield to.
It sounds like that would be,
yeah, right. And so we are doing that, but just for this, we're only taking this data from plots that do not have nitrogen added. Because also like additional nitrogen can influence decomposition dynamics and microbial activity. Yeah. Oh yeah. We're taking all that into account. Yeah. And so and we statistically account for all of that. So yeah. So right. Exactly. So I see what you're saying. So you can also just present the results separately and say, if you don't add any nitrogen fertilizer and you just do cover crops, you can get
Be
this.
an added benefit, possibly. You're not adding another fertilizer.
Yeah,
The growth.
yeah. But at the same time, if you're not adding another fertilizer to that growth curve, I think that you will see you will have yield penalties, because if you look at the numbers, I mean, you're not getting that much nitrogen, not all the time. I think the take home for me here was some years you're going to get maybe more synchrony and some years you're not. I don't think it's real. I think at least for what we found in the first transitional parts where we had a conventionally managed system and then we're starting to use cover crops, we're not finding consistent results to add that show that these cover crops are giving us enough nitrogen during the growing season to offset fertilization. And maybe that'll change the longer you use cover crops. And that's only the second year. If you look at the first year, it's different. And so I think it actually really depends on the year and the weather conditions. I think it's a lot. You lose a lot more in the second year than in the first year. Yeah. And so I think that again, it depends on the weather. And so it's not necessarily like this reliable thing where you're going to be able to get the same results every year. Yeah. And so just depending on cover crops to provide nitrogen in the scenario where you're planting cover crops in the late summer or fall, and you're hoping, oh, well, I'll get in there and I'll plant something in the spring and I see the residues and they're decomposing. So surely I'm getting this much nitrogen, especially when you look at, there was a slide I had, the promised nitrogen from those crops, and you think, okay, well, I have this much biomass from these crops, that should translate to this much nitrogen. Surely I'll get it in the spring. And at least from my research, it shows that not necessarily, so some years you might get more, some years you might get less, but you're actually losing a lot before you can even get in there. Not to say that there aren't other benefits, but in terms of, can you reduce your nitrogen fertilizer if you're using these cover crops for especially, and again, the caveat too is these are the first 2 years of transition from conventional to organic to this cover cropping system. If you're cover cropping heavily and you have and you're doing this for 5, 6, 7 years, the results might look different, and that's also something to consider. So the last thing I'll talk about is the impacts of decomposing cover crops on soil biology. So this is something that go ahead.
Good question. In between garlic harvest and cover crop planting years, were you just keeping the field fallow? Or was it
buckwheat? We just had buckwheat in it. So we planted buckwheat pretty much as soon as the garlic was out, and then we terminated the buckwheat. That's a good question. Oh, I'll talk closer to the question was if we had the field fallow between garlic harvest and planting garlic, and we had planted buckwheat so in July and then terminated about a week before we planted our cover crops. Okay, so the last thing I'm going to talk about, and I don't have all the results in from this yet, was one of the things that we were interested is the impacts of decomposing cover crops on soil biology. So as I was taking those samples, the cover crop samples from the field, I was also creating that growth curve, and I was also taking soil samples, and I was using those soil samples to look at soil nitrate. I'm going to look at the active carbon, as I mentioned. I also looked at microbial respiration to see if microbial respiration was different among the decomposing cover crop treatment, and also the soil food web, which I'll talk about in a bit. So the idea was that we have increased organic. We know that factors that include reduced soil disturbance, increased organic inputs, can cause changes in microbial populations, which in turn can change decomposition and nutrient cycling. So that's the reason we wanted to look at these things in the first place. And first and foremost, we looked at soil respiration. We wanted to know if the decomposing residues of fall planted annual cover crops can impact microbial respiration in the spring. And so as you can see, this is the curve we got, which looks like something out of a textbook, and the answer is no. For us was that the decomposing cover crop residues did not have a significant impact on microbial respiration. What did have a significant effect on microbial respiration was the time that we sampled. So we sampled every 2 weeks, and you see this curve. So you see these fluxes in microbial population that are most likely caused by weather fluctuation. So what those results mean is we're still parsing this out because we're still looking at nitrate and soil active carbon. Was that 1, 2 years might not be enough to see changes in microbial populations in a field. 2, this amount of residue might not be enough to see changes. Or 3, seasonal fluctuations are just always going to be stronger than the amount of residue we're putting in. And again, there's a lot of unanswered questions here. And so I'm still interpreting these results. So I guess I can't speculate too much, but I think this is an important result because to show like sort of the timeline and what we can expect in terms of microbial activity and changes that like the management changes, how they impact soil microbes. Because we like to talk about how soil microbes change with these management strategies. And at least what I found is that in the first 2 years, you're not seeing these significant changes. And so the last thing I'm going to look at is the soil food web, and I'll talk to you about how I'm going to do that in a second. The soil food web, just a brief introduction, it documents the relationships between the complex community of organisms and environmental processes that exist in the soil. And these organisms redistribute minerals through space and time. What that really means is that, as I talked about before, you have different nitrogen dynamics. These organisms can ingest, for instance, nitrogen, move around, and then excrete it somewhere else. That's how they're distributing minerals through space and time. Again, if nitrogen, for example, using nitrogen as an example again, is incorporated into microbial biomass and then released later, then like when the microbe dies, again, it's just distributing that mineral through time. So that's what space and time means. Sounds a little bit futuristic. Organisms within the soil food web also directly influence soil fertility through the mineralization and mobilization of important plant nutrients. They decompose organic matter, modify soil structure, and impact aggregation, regulate pest populations. So if you have a balanced food web, then hopefully it's regulating itself. You don't have these explosions of pest populations, and they sequester carbon. Okay, so the soil food web, I'm sure that probably most of you have seen this diagram before. How does carbon enter the soil food web? And first and foremost, through plants, producers, and their residues. So as I mentioned before, 30% of the photosynthetics that plants produce are exuded through their roots and go directly into the soil food web. Also organic matter decomposition. So residues of things like cover crops decompose, and carbon enters the food web through the rises, through these decomposing residues. And then from that, the carbon continues to move through the soil food web. So you have every single trophic level, you have the amount of carbon is decreased. So as you can imagine, so the first level, things like nematodes, fungi, bacteria consume the carbon. Then those things are consumed by things like arthropods, again, more nematodes, protozoa, consumed again by arthropods, nematodes, and then by macrofauna, birds and animals. And so as you can see, with every trophic level interchange, the amount of carbon decreases. So in order to get a structured and complete and I guess healthy food web that can support all of these levels, you need a lot of input at the first level, which is why things like cover crops and keeping plant roots in the ground and having decomposing cover crops can be important for the soil food web and why we're looking into it. And so the way we're doing it is through something called nematode faunal analysis, where we're using nematodes as biological indicators. And the reason that we can use nematodes as biological indicators to look at soil food web status is that if you noticed here, they're actually present in every single trophic interchange. They also have a short generation time. They respond relatively, not all of them, but they tend to respond quickly to changes in management and into inputs. And also they're easy to identify. Well, I wouldn't say easy, but you can identify them by their morphological features. So that's relatively easy in terms of it's cheap. And if you know how to do it, you can just basically look at what the nematodes are doing in the soil food web based on their feeding habits. So what you see down below is the feeding parts of nematodes. And so if you can see the differences, those are, A is a bacterial feeder. It has a little cup. And so that's where bacteria enters, or soil solution enters, and it consumes bacteria. The second one, B, is a fungal feeder. It has this little needle thing at the top. That's a spear. What that does, that punctures hyphae and extracts things from it basically feeds on fungal hyphae. The third one is a plant parasite, so that can feed on plant roots. It has a stylet as well, but you see it has these little muscles at the bottom and needs to be a little bit stronger to puncture a plant root than to puncture hyphae. And then the last two are a predator, eats other nematodes, and an omnivore, which has that big spear-looking thing at the top. So basically, by looking at these feeding habits, we can infer what is happening in the soil food web, because as you can imagine, if there's an explosion of bacteria, there's an explosion of bacterial feeding nematodes. And so by identifying and counting these nematodes, we can figure out what's going on. And so for those of you who don't know, because this is a question I get asked a lot, are what are nematodes? I think you'll be shocked to find out that they're the most abundant form of multicellular animals on Earth. They're literally everywhere. 4 out of 5 animals is a nematode. And they're found in all terrestrial and aquatic habitats, so rivers, lakes, streams, soil. And they're typically 1 to 2 millimeters long. Most of them are microscopic, but they do range from 100 micrometers to 8 meters in length. The largest one is 8 meters is Placenta nema. It's found in the placenta of whales. But if you want to see a nematode with the naked eye and you don't want to, if you're not interested in whales, if you buy salmon or cod from a grocery store and you season it with some salt and you start to heat it up, you'll often see a word crawling out. That's a nematode. But yeah, they really are. They really are everywhere. And so, yeah, again, because there's so many of them, 4 to 5 animals, as I mentioned, there's tons of them in soil, and they're actually performing a lot of different functions. For instance, bacterial feeders, when they ingest bacteria is generally very nitrogen rich. It's more nitrogen than bacterial feeders need. So they excrete that nitrogen and they make it more available for plants. So that's one of the ways that we can kind of use nematodes to look at nutrient cycling in soil food webs. So free living nematodes are nematodes that are non-parasitic to plants and animals. So you might be familiar with parasitic nematodes, things like root knot nematode, which affects like carrots and peppers, has a wide host range, or stunt nematode. Some of the parasitic nematodes are more well known because they cause significant crop loss. The free living nematodes are non-parasitic and they're still very abundant. They feed on bacteria, fungi, algae, and other nematodes, and they're found in all ecosystems on Earth and on almost all trophic levels, which is what makes them really useful as bioindicators. And so what I'm doing is I'm basically extracting these nematodes from the soil samples. The way I do that is I extract them in water. It's relatively easy. It's something that you can actually do at home, but you need a microscope to mostly see them. If you take a pie pan and you cut out the middle and you put a mesh screen and then you put a dairy filter on top, you put some soil on, fill it up with water, and you have a pie pan underneath it's basically sitting in water. The nematodes within 24 hours will swim through the dairy filter, and then you'll have basically a water solution filled with nematodes. This is gross things. Yeah. Yeah. So anyway, so what I do is I extract these nematodes, I look at them in the water, and then I identify them to family, and I then count the different nematodes in different families, and then I can make some assessments about the soil food web. So I'm still doing that. I have hundreds of samples, and I think I'm going to be done with all of them in about a month. And just to give some background on how it works, so you have these different nematodes that can be separated on this, what we call a colonizer persister scale. And what that really means is we have some nematodes, like bacterial feeders particularly, they're opportunists. So if you have this flush of bacteria or like nitrogen or something in your, if you have a flush of nitrogen and you have a lot of bacterial decomposition happening in your soil, then these bacterials are going to be there. They don't really care about disturbance. They're not sensitive to disturbance, and they're going to explode in population. They have a lot of young. They have a really short generation time, something like 1 to 2 weeks, and you're seeing this explosion of bacterial feeding population. On the other end, you have nematodes like the omnivores, which are really large. They have thick cuticles, which is basically like their skin. They only produce like 1 or 2 young, and they have a year. Some of their life cycles are about a year, and so they're really sensitive to disturbance, and they only occur in these really stable food web when you're not getting a lot of disturbance and when things are pretty structured. So by looking at what these communities look like, we can kind of infer what a food web looks like. And so these are just some of the things, the calculations of the conditions I'm going to take. So these are, might not be that interesting to you, but these are the things I'll be able to tell from these populations. So enrichment and the maturity index looks at ecological succession. I'm not expecting to see many changes there from like a year of cover cropping, but that's the response of environmental changes. The enrichment index and the channel index, I think, are probably the most interesting. The enrichment index is the assessment of food web response to available resources. So again, how do food webs respond to different cover crop or resources and like different compositions of cover crops? And the channel index, which reflects the primary decomposition channel that's taking place, whether it's fungal or bacterial in the food web as the resource availability shifts in the system. So in the end, we'll be able to put it on a plot like this. Again, I'm not going to talk about this too much, but if you're interested in this, I can talk to you about it at length at another time, or just email me and we can chat. And so again, in progress, I'm going to have nematode faunal analysis to wrap this up, the changes in active carbon, and the nitrogen use efficiency. So that's all I have for you today. If this is my email, if you want to get in touch with me about anything, please feel free to